the amplification of DNA actually happens in this appliance, okay? As you can see here, we have the different steps of DNA amplification, okay? Here we have the denaturation step of our thermocycler plant, okay? At this step, your DNA gets denatured. By denatured, I mean that your double-stranded DNA, okay, by means of high temperature, which is 95 degrees centigrade, for 5 minutes gets denatured, and the two strands of it get detached from each other. And now we have a single-stranded DNA, okay, which we will need for next steps. This steps only happens for the first time. In the next cycles, we have it for 45 seconds, okay, not like 5 minutes anymore, okay, it would be way too time consuming, okay. The next step of it, at which the primers, the forward and reverse primers, get attached to your sequence, a specific sequence in your genome, is here, okay, which happens about 60 degrees centigrade, and it lasts for 45 seconds. Imagine it is your whole genome, okay, which has become single-stranded, okay. Here we have a specific sequence of it which we want to get amplified, okay, which we want to detect, okay. The forward and reverse primers gets attached to it, okay, and they try to get that specific sequence amplified. Okay, that amplification actually happens here in the fourth step, which is happening here for 45 seconds at 72 degrees centigrade. Okay, after all these steps are done, okay, the cycles get repeated. Okay, here for this plant, the cycles get repeated for 36 times. Okay, then we have our PCR product. Okay, what we need to do next would be doing gel electrophoresis, okay, so that we check our DNA product with the positive control, okay. Our DNA product is charged negatively, okay, and we put it in a negative field, okay, and from a negative pole of our electrophoresis duct, okay, it gets pushed away, okay, and they get separated, you know, according to their size, okay, and then you have your positive control, and then you check the bands and you can confirm whether you had that specific DNA in your sample at first or not, okay. So everyone, please make sure that you have taken a good look at all these steps of this thermocycler, and also it would be better if you took pictures of it, okay. It would be needed for your exam, and also the different steps of it and what happens in each of them also matters. So is there any question?
Thank you very much. So all of you can see the steps and the machine and how it works, and after that was you come here. You see? No? All of you see it? So if you suppose one stay there.